It is estimated that there are between 10 million and 100 million different species. Despite this wide variety of organisms, it remains difficult to define what it means to say something is alive. Which of the following can be described as the smallest living unit? DNA, cell, organelle, or protein. Now, if any of you got this question wrong, I just jump out of the window. Which is correct? A, B, C, or D? All together? Come on, all together? B. B?
B failed.
On top? Okay. Yes, of course it's B. Of course it's B. 1.5. The flow of genetic information is controlled by a series of biochemical reactions that result in the production of proteins, each with its own specific order of amino acids. Choose the correct series of biochemical reactions from the option presented here. Now this question, I find it's a little bit tricky. I'll explain to why later. Replication, transcription, translation. Replication, translation, transcription. Translation, transcription, replication. Translation, replication, transcription. A, B, C, or D. Who said A? Raise your hand. What did you say?
B.
This is not correct. The right question was, the right answer was A. Replication, transcription, translation. Now, why this question is tricky? It's because replication, it's something different. Replication doesn't make protein, okay? Replication is just copying DNA, okay? But first you copy the DNA to make more cells, then you transcribed RNA from DNA, and then you make translation, so you make protein from transcription. So from protein from RNA. So the right answer is A, indeed. But replication is completely different from making proteins or from making RNA. Okay, it's just duplicating DNA. So it's a little bit tricky. Question 1.11, which is a little bit more difficult, this question. Which statement is not true about the events' conclusions from studies during the mid-1800 surrounding the discovery of cells? A. Cells came to be known as the smallest universal building block of living organisms. B. Scientists came to the conclusion that new cells can form spontaneously from the remnants of ruptured cells. Light microscopy was essential in demonstrating the commonalities between plant and animal tissues, or new cells arise from the growth and division of previously existing cells. A, B, C, or D? Who said A? Raise your hand. You said A. This is not correct, okay? Yeah, this is correct. This is actually correct. Why do you think it's not correct?
Cell is cell. Cell came to know as a cell.
Yeah, cells came to be known as the cell, but they observed in the microscope that everything that is alive is made of small cells. So they thought everything is made of cells. So this is the smallest universal building block of living things. If it's something that is living, it's made of cells. So A is actually correct. So everything is made of cells. So cells is the unit of living things. Who said B is not correct? B is the right answer. Yeah, B is actually the correct answer. Scientists came to the conclusion that new cells can form spontaneously from the remnant of ruptured cells. No, of course not. When the cells are dead, the cells will stay dead. Okay? No spontaneous generation can happen. However, this is very interesting that before scientists started to study living things, they thought cells could spontaneously come alive. That was an interesting thing, is that if you had a pot containing some substance, and then after some time, they were observing cells, like bacteria and things, and they thought, this is God making cells all the time. They thought it was like this. But of course, then clever people noticed that if they put a cover, there are no cells coming. So these cells come from something else. So anyway, this is just for the little history. So of course the right answer for 111 is B. So this is not true. 112, A, B, C, or D. What unit of length would you generally use to measure a typical plant or animal cell? Do you measure a cell in centimeter? Do you measure a cell in centimeter? Well, it depends which cell you are talking about. Which is the most common unit of length associated with cells? Centimeters, nanometers, millimeters, or micrometers? A, B, C, or D? Who says A? Who says B? Wrong. Did you say this is a very, very small cell? Nanometer. Extremely, this is the size of a nucleus, so it's too small. It's too small. Who says C? Who says D? Yes, D. Cells are usually under the order of micrometers. Even bacteria is micrometers. However, viruses, they go down to the nanometer and stuff like that. So, nanometer is more like the order of molecules and stuff. Okay, have a seat, take your questions. We have only started. And we have reviewed question 1. This is correct. Question 1.5. This is correct. 1.11. This is correct. 1.12. Interesting. Like this gentleman. You are not correct. It's micrometer. A cell is about micrometer. The length of a cell is in micrometers, not nanometers. Okay? 117. By definition, prokaryotic cells do not possess. So this is also one of the questions that I jump out of the window if something is not correct. So, A nucleus, B, replication machinery, C, ribosomes, D, membrane bilayers. By definition, A, B, C, or D. Who said A? Did you say, what did you say?
C.
Did you say C? No, this is not correct. Okay? This is not correct. So, prokaryote means before the nucleus. This is what it means. So by definition, pro means before, and karyote comes from the Greek karyon, and this means nucleus. So by definition, they are before the invention or the evolution of the nucleus. So A is the correct answer. So you should never say something you are not able to carry on like I did not jump out of the window. Okay. Question 119. indicate whether the following statements are true or false. Now, these questions, I like them because only one very little word can make the question true or false. A. The term prokaryote and bacterium are synonyms. Is it true or false? Now, this is really tricky because it's also English. It's not only biology, but it's also English question. Who said this is true? Who said this is false? This is false. This is false. Why it is false? Why it is false?
Prokaryotes didn't have nucleus, but bacteria have nucleus.
This is not true. Why is it not true? Why is A not true?
Prokaryotes includes archaea.
Exactly. So prokaryotes is a big group of cells that, yes, contains bacteria, but they also contains archaea. So they are not synonyms. It's just like saying mammals and eukaryotes are synonyms. This is not true, right? So this is the same. So bacteria have a nucleus. They have no nucleus. Okay. Bacteria have no nucleus. They are part of the prokaryote. So. Be careful. Bacteria do not have nucleus. So this is why they are part of the prokaryote, but they are not synonyms of prokaryote. What about B? Is it true or false? Who said false? Who said true? Yes, very nice. C. Is it false? Raise your hand. Is it true? Raise your hand. It is true. Yes. Why is it false?
I think no prokaryote has cell walls. Prokaryote doesn't have the cell walls.
C no. C no. Hey, don't worry. Don't worry. So, yes, bacteria have cell walls. Just like plant cells as well. So don't worry. Never be ashamed of your answer. That's okay. That's okay. We are all learning. So B and C were both true. Okay, so we are reviewing the question. And take a breath. And then, of course, this is correct. This is correct. This is correct. This is not. Yes, micronutrients. Yes. And this is correct. And it should be A is actually false.
Oh, it's false.
It's false because prokaryotes contain bacteria and archaea.
Okay.
So it is not really a synonym.
Right.
It's just like saying mammals and eukaryotes is synonym.
I did write the text in case.
Okay, so be careful. This is actually false. A is false. But B and C is true. Then 121. Indicate whether the following statements are true or false. If the statement is false, explain why it is false. A. Who said it was false? Who said it was true? Excellent. B. False. Raise your hand. It is false. It is false. Why it is false? Mitochondria are thought to have evolved from anaerobic bacteria. So this is what? 2 letters. Only 2 letters make this sentence false. Only 2 very small letters. Anybody, please explain to me why this question is false. And to the people who said it was true, explain to them why it is not true. Anybody?
It is false because mitochondria from aerobic bacteria.
Exactly. Mitochondria, just because they use oxygen to power their metabolism, they are thought to have evolved from aerobic bacteria, not anaerobic. C. Photosynthetic bacteria contain chloroplasts. Who said it was true? Who said it was false? You are right. It's actually false. Photosynthetic bacteria contains no chloroplasts. Plants contain chloroplasts. But just like bacteria do not have mitochondria, right? So photosynthetic bacteria do not have chloroplasts. Okay? Chloroplasts plant cells have. Algae have chloroplasts. But bacteria, even when they are photosynthetic, they do not have chloroplasts.
How do they then do the photosynthesis?
They contain proteins that do the photosynthesis, but they are not included in chloroplasts. What is thought, and maybe why you may be confused, is just like an animal cell containing mitochondria is thought to originate from eukaryotic cells is thought to originate from a symbiosis between an ancient cell who could not use oxygen for its metabolism and a bacteria that could use oxygen and that became later mitochondria. The same is true with plant cells. Later on, when this cell developed and evolved and had mitochondria, then it is thought that it became a plant cell by getting a bacteria that could do the photosynthesis that later became a chloroplast. But these bacteria, still now, these bacteria are still alive now, but they have no chloroplast. Later they may have become chloroplast when they did the symbiosis with the cell. But currently, the bacteria that are doing photosynthesis do not contain chloroplasts. Only plant cells do. But they have proteins that can do the photosynthesis. Okay. 122. Some prokaryotes can live by utilizing entirely inorganic materials. Which of the following inorganic molecules would you predict to be the predominant building block for fat, sugars, and proteins? Now, I looked at the answer of some people last week, and I noticed that some of you understood that you had to say which is the main building block for fat, which is the main building block for sugar, which is the main building block for proteins. Actually, which is the main building block for all three, not separately. Okay. Like for example, some of you may have said, for protein, this is N2. For fat, this is CO2. For sugar, this is O2. This is not correct. Okay. Which is the main building block for all of these three organic molecules? For fat, for sugars, and protein. Which do you think is the main building block? A, B, C, or D? Who said A? Who said B? Who said C? Yes, C. Why? Why CO2 is the main predominant source? Why do you think it is the main source of all these organic molecules? What is the justification for that? Because they are nitrogen also exists in protein, right? Proteins are full of nitrogen, and DNA is also full of nitrogen. So, but why did you choose CO2? Because it contains carbon, and carbon is the most frequent atom in our body. So this is CO2. Okay, 123. So normally, I will just say, okay, A3, B5, C1, D2, E4, F7, and G6. Okay? That one was very easy. All right. 124. Fill the blanks. Eukaryotic cells have a bigger and more elaborate, are bigger and more elaborate than prokaryotic cells. By definition, all eukaryotic cells have a nucleus, usually the most prominent organelle. Another organelle found in essentially all eukaryotic cells is the mitochondrion, which generates the chemical energy for the cell. In contrast, the chloroplast is a type of organelle found only in the cells of plant and algae and perform photosynthesis. If we were to strip away the plasma membrane from a eukaryotic cell and remove all of its membrane and closed organelles, we would be left with the cytosol, which contains many long, fine filaments of proteins that are responsible for cell shape and structure and thereby form the cell's cytoskeleton. Okay, so I try to organize these questions so that what information you have in one question, you can use it to answer correctly other questions. So for example, 124 and 121 had a link. Because when you say 121, photosynthetic bacteria contain chloroplasts, then if you have in 124, if you correctly answer the question, in contrast, the chloroplast is a type of organelle found only in the cells of plant and algae. Then, of course, so you can use this, but yeah, maybe. All right. 126, which is the correct word to put in the blank? A, B, C, or D? Who said A? What A? Who said B? Who said C? Who said D? Ribosome. This is not true. Where are the ribosomes? Where are the ribosomes? Where are they? The ribosomes. They are in the cytosol. The ribosomes are the endoplasmic reticulum in the cytosol, in the cytoplasm. Okay, so the right answer for this is the chromosome. The chromosome is the complex of DNA and the protein packed up into the nucleus. So of course it is the chromosome. 136 was maybe a little bit tricky. A, DNA, this is easy. So they all should be okay. Okay? Animal, plant, bacteria contain DNA. Nucleus, only animal and plant. Bacteria have no nucleus. C, plasma membrane. Animal, plant, and bacteria have plasma membrane. D, chloroplasts. Only plant. Okay? Cell wall, plant and bacteria. Lysosome, animal, plant, and bacteria. And this is correct. It's not correct. Lysosome, animal and plant. No bacteria. Okay. Lysosome, animal and plant. Mitochondrion, animal and plant. Golgi apparatus, animal and plant. So essentially bacteria do not have any organelles. Okay. And lysosome, mitochondrion, and Golgi apparatus, they are all organelles, as well as nucleus and chloroplasts. 149. Rate the following things as good or bad processes to study in the model organism E. coli. Let's say you are a scientist and you want to study these things. Would you use E. coli or not? What about A? You want to study the formation of the endoplasmic reticulum, how it is regulated, how is it formed by the cell. Is it good or not to study bacteria to understand this process? Who said it was good? Who said it was bad? Yes, it is bad. Because bacteria do not have endoplasmic reticulum. B, DNA replication. You are studying how DNA replication is regulated. Is it good to use bacteria or not? Who said it was bad? Who said it was good? Yes, everybody, nice. C, how the actin cytoskeleton contributes to cell shape. Who said it was bad? Who said it was bad? Yes, you are right. You are right. It is bad. Why it is bad? Why it is bad to use bacteria to study the cytoskeleton? The actin cytoskeleton. Because the cytoskeleton of bacteria is completely different from the cytoskeleton of eukaryotic cells. Okay, so it is not good at all. The organization is completely different. Actually, bacteria hardly have any cytoskeleton, even less active. Okay, so this is bad. D, how cells decode their genetic instructions to make protein. Is it good or is it bad? Who said it was good? Yes, it is good. It is good because how the bacteria make their proteins is quite similar to how eukaryotic cells also make their protein. Although it's a little bit different because they have no endoplasmic reticulum where the ribosomes are, but they do have ribosomes. Okay, so it's still okay. Good or bad for the E1? How mitochondria get distributed to cells during cell division? Is it good or bad? Who said it was bad? Yes, it is bad. Because bacteria do not have mitochondria. Right. So A, bad, B, good, C, bad, D, good, E, bad. 159. You wish to explore how mutations in specific genes affecting sugar metabolism might alter tooth development. Which organism is likely to provide the best model system for your studies and why? Who said it was A? I'm saying it's A. I'm saying it's A. Horses have beautiful teeth, right? And it's very important for them to eat because they eat grass and stuff. Why have you said something else? Why have you said maybe B? Most of you have said B. Yeah? You have said B. But I'm saying it's A. Why are you saying B?
Because horses are slow to reproduce.
Exactly. Exactly. And they are huge. So if you want to study 100 horses and try to make transgenic horses, it will take a huge amount of money and space. So you are right. Of course, mice is the best because they have teeth and they can reproduce easily, and you can actually make genetic modifications pretty easy. All right. 165. This was more of a writing question. I will read it completely. The antibiotic streptomycin inhibits protein synthesis in bacteria. If this antibiotic is added to a culture of animal cells, protein synthesis in the cytosol continues normally. However, over time, the population of mitochondria in the cell becomes depleted. Specifically, it is observed that the protein synthesis machinery inside the mitochondria is inhibited. A. Explain this observation based on what you know about the origins of the modern eukaryote. How can you explain this? This is a more difficult type question. So you are studying this antibiotic that you don't know why, but it inhibits only the protein synthesis in the mitochondria. But the cell is okay with it. The cell can still synthesize its own protein. So, how can you maybe more or less explain how could this happen? And there is a big hint, right? It's like, based on what you know about the origin of mitochondria. Okay, so you know bacteria are thought to be, some bacteria at least, are thought to be the ancestor of mitochondria, right? As I said, I explained before, it is thought that a cell without mitochondria, a kind of prototypical eukaryotic cell, got into a symbiosis with a bacteria that later became mitochondria. Okay. And here you are using an antibiotic that doesn't do anything for the protein synthesis inside the cell, but it affects protein synthesis inside the mitochondria. Now, I told you during the lecture that mitochondria have their own genome, right? They have their own genome. So it means they are synthesizing their own proteins. Okay? They are mitochondrial proteins that are encoded by the mitochondrial genome. So how could you explain that this antibiotic can only inhibit the synthesis of the protein into the mitochondria? What's the explanation?
This streptomycin inhibits some proteins, and these proteins are only used in bacterial DNA transcription.
No, it could be transcription. In this case, it's protein synthesis, so it's translation. But essentially, you are right. So what happens is that the machinery that makes protein in the cytoplasm and the machinery that makes protein in the mitochondria is completely different. They are completely different proteins. The proteins that synthesize, like a ribosome and all these things, that synthesize protein in the mitochondria are completely different from the one that are used by the cell. And the ones that are there in the mitochondria, they are very close to bacterial machinery. So because they are different, you can have antibiotics that specifically inhibit the protein synthesis in bacteria, but not in eukaryotic cells. So when you use these antibiotics on eukaryotic cells, the eukaryotic cells are okay with it, but the mitochondria protein synthesis machinery, who is very close to that of bacteria, is inhibited. Okay, so this is what's happening. And then, what would you expect to observe if, in a new experiment, animal cells are treated with this diphtheria toxin? It's a compound that is known to block cytosolic protein synthesis, but does not have any impact on bacterial growth. So for the same reason, you can make or you can discover an inhibitor of protein synthesis only in the cytoplasm. But mitochondria will be alright. So what data will you get? What kind of results will you get? What would you observe? If you inhibit the protein synthesis in the cytoplasm of the cell. And then, so you stick on your cells this inhibitor, and then you take a look at your cells the next day, the day after, 1 week later, what are you going to see? Can you sort of imagine? What is going to happen?
Cells will die,
Exactly.
or even probably breaks up.
Yes, exactly. So after 1 day, maybe the cell will still be okay, but the amount of protein inside the cell will go down very quickly. But the cell cannot survive without its protein. It must make new protein all the time. So if you always inhibit protein synthesis in the cell, then the amount of cells will go down and down and down, they will die, and at the end you have no more cells. You will have no more bacteria, you will have no more mitochondria either, because mitochondria depends on the cell to survive. Okay? So if you kill the cell, mitochondria will also be killed. But immediately, only the protein in the cell will decrease, and then this will lead to cells being killed. Okay? So it's very dramatic. But then I go back to the A. Would you expect to use this antibiotic that only kills protein synthesis in mitochondria? Would you expect this to kill the cell? B, yes, it will kill the cell. You will completely eradicate proteins in the cell, so then the cell will die, unable to survive. But then, let's say you, for 2 weeks, for 3 weeks, you always give the cell this protein synthesis inhibitor in the mitochondria. So let's say after 3 weeks, mitochondria will have disappeared. Will the cell survive?
I think no.
No. I say yes. I say yes. But only on some conditions. These cells, actually, I find it's really amazing. I want to start studying these things. These cells are called I don't know why, but they are called rho. This is the rho Greek letters, rho 0. And these cells have no mitochondria. They have no mitochondria, but they survive. And you can do these rho 0 cells with any cells. You can take mammalian cells, you can take your human cells, or you can take mouse cells. You put inhibitors of synthesis of protein synthesis in the mitochondria, and then at the end you have cells without mitochondria. But they survive only on some condition. Do you guess? It's more difficult. What might you do to allow the cell, even though they do not have mitochondria, they will be able to survive? Can you kind of guess what you could do to allow the cells to survive? Based on the function of the mitochondria. Okay, so this is more difficult, but mitochondria are very critical for the metabolism of sugar, right? You get energy from the sugar, but eventually it goes into the mitochondria where you make a lot of energy. So you must provide into the culture medium these things that provide the cells with energy. The cell will not be able to make these because they have no mitochondria. But if you provide these energy-giving substances, they will be able to survive. Moreover, many molecules in the cell, in the eukaryotic cells, they are made in the mitochondria. Like, for example, nucleotides. Some nucleotides, they are made into the mitochondria. Even though they are used by the cell depends on the mitochondria to make these nucleotides for itself. For example, uridine. You know uridine? This ACTG, this is in DNA. but ACU. G, this is in RNA, right? Uridine is actually made into the mitochondria. So if you break the mitochondria, the cell will not be able to make uridine. So you must put into the medium uridine, so that the cell will be able to grow with uridine, even if it doesn't have mitochondria. So all these things can happen. But the cells are happily, they survive happily, and they grow, and they can multiply, as long as you give things that used to be provided by the mitochondria. So it's a little bit more complicated. Okay, question 210 is a bit more easy. Which of the following elements is least abundant in living organisms? This is easy. A, B, C, or D. The least abundant. Everybody together? A. A, yes. Yes, of course. 215. Indicate whether the statements below are true or false. If a statement is false, explain why it is false. Which is the only one that is false? The only among A, B, C that is false is who said B is false? B is actually true. B, A, C, O, and N are the most common elements in biological molecules because they are the most stable. Who say it's true? Ah, nice. Who says it's false? Yes, of course it's false. Yes, nice. C. Some atoms are more stable when they lose 1 or 2 electrons, even though this means they will have a net positive charge. Is it true or false? Who say it was true? Yes, nice. It is true. It is true. Okay, number 216. A covalent bond between two atoms is formed as a result of the A, B, C, or D. Everybody together. A. A. It's the sharing of electrons, of course. Then 217, an ionic bond, everybody together, is
D.
D. Yes, it's D. Transfer of electrons from one atom to the other. 218, I will read the whole thing. Whereas ionic bonds form a salt, covalent bonds between atoms form a molecule. These covalent bonds have a characteristic bond length and become stronger and more rigid when 2 electrons are shared in a double bond. Equal sharing of electrons yield a non-polar covalent bond. Non-polar covalent bond. If one atom participating in the bond has a stronger affinity for the electron, this produces a partial negative charge on one atom and a partial positive charge on the other. These polar covalent bonds should not be confused with the weaker non-covalent bonds that are critical for the 3-dimensional structure of biological molecules and for interactions between these molecules. All right. 227. Which of these molecules contain polar bonds? Molecular oxygen, methane, propane, or water? Which contain polar bond? Water. Yeah, this is D. This is D. 233. The pH of an aqueous solution is an indication of the concentration of available protons. However, you should not expect to find lone protons in solution. Rather, the proton is added to a water molecule to form a hydroxide, ammonium chloride, or hydronium ion.
Hydronium.
Hydronium. D. Hydronium. D. 224. It's about cell membrane. So is it A, B, C, or D? This should be easy, right? This is probably all together. Which is correct? A? Who said A? Who said B? Yes, it is B. It is B. That being said, if there had been A, B, C, D, E proteins, that would have been more difficult. Because proteins are also needed to make this permeability of the membrane, because you have channels, you have transporters, but sugars, amino acids, and nucleotides, they are not important at all in making the barrier. So among these answers, the best is B. 247. Cells require one particular monosaccharide as a starting material to synthesize nucleotide building blocks. Which of these monosaccharides below fills this important role? Glucose, fructose, ribulose, or ribose? This should be probably everybody is correct there. Who said D? Yes. This is D, of course. 254. Many types of cells have stores of lipids in their cytoplasm, usually seen as fat droplets. What is the lipid most commonly found in these droplets? A, B, C, or D? D. 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 Cholesterol is an integral component of the cell membrane, but it is the storage droplet, especially in the liver, is D, triacylglycerol. 255. Choose the answer that best fits the following statement. Cholesterol is an essential component of biological membrane. Although it is much smaller than the typical phospholipids and glycolipids in the membrane, it is a Nantoka molecule having both hydrophilic and hydrophobic region. Amphipathic is the right choice. Amphipathic. Why? Why is it not hydrophobic? Why is cholesterol not hydrophobic? If you remember. Why is cholesterol not hydrophobic? Like the tail of the phospholipid. Who actually wrote D? Yes. Why did you write D? Why did you know cholesterol is amphipathic? Did you just remember, or do you see the structure of cholesterol in your mind?
I remember that it means like both hydrophilic and hydrophobic.
Yes. So this is what it means. They are both together, right? But any of you kind of know why? Because cholesterol, it's made of hydrocarbon chain, with many cycles and everything. But on one end, it has OH, and that is enough to make it amphipathic, because this OH will make hydrogen bonds with many things. But the whole rest of the molecule is just a big hydrophobic thing. But yes, you are right. It's amphipathic, so that's nice. 256. I will read the whole thing. Proteins are polypeptides built from amino acids, which each have an amino group and a carboxyl group attached to the central alpha carbon. There are 20 possible side chains that differ in structure and are generally referred to as R. In solutions of neutral pH, amino acids are ionized, carrying both a positive and negative charge. When a protein is made, amino acids are linked together through peptide bonds, which are formed by condensation reactions between the carboxyl end of the last amino acid and the amino end of the next amino acid. Okay. These questions are very easy. 257. I've already answered this one. Which is the correct one? A, B, C, or D altogether?
B.
D. B. B. B, 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 B. B. B like baby, okay? It contains thymine. 261. Both DNA and RNA are synthesized by covalently linking a molecule nucleoside triphosphate to the previous nucleotide, constantly adding to a growing chain. In the case of DNA, the new strand becomes a part of a stable helix. The two strands are complementary in sequence and antiparallel in directionality. What is the principal force that holds these two strands together? A, B, C, or D? So this also should be B. B. I like the 268. It's a very long question that takes a long, precious time to read. Okay? So, are you going to bet? Or are you going to not bet? Do you think that the protein that your friend wants to make will work or not? So are you betting that it will not work? Or are you betting that it will work? Do you think it will not work? Yes. You are betting that it... Who says it will work? Who says it will not work? Yes, okay, you are all correct, so I will not explain this one. Okay? It will not work. 3-1. A, B, C, or D. Metabolic, catabolic, anabolic, or biosynthetic.
This question asks reason.
Ah, all right. Yes. What is the reason? Why will it not work? Why will it not work? If you make a protein that has exactly the same sequence of amino acid, but in the completely different order. Not only in a different order, but completely opposed order. Why will it not work?
The copy has mirrored conformation, so it binds other molecules differently.
Yeah, not only so each amino acid has a polarity, right? There is the carboxyl end and there is the amino end. But if you make a protein that has the same amino acid but it completely the different configuration, not only the way the protein will fold and the way all the non-covalent bonds will be formed between amino acids will be completely different. So the three-dimensional structure, the tertiary structure of the protein will be completely different because the relationship of each atom with each atom will be completely different because of this polarity, okay? Because of this polarity. Three, one. This is, which one is correct? A, B, C, or D? That's very easy, this one. The specific subset of these reactions that break down nutrients in food can be described as breaking down is A, B, C, or D. Who said A? Who said A? Who said B? Yes, it is B. Catabolic. You are half right. Why are you half right? You are not completely incorrect. You are 50% correct. Why you are 50%? Why do you think you are 50% correct? 50% of your answer is correct. Why? Because A, metabolic, it's actually B plus C. Metabolism is anabolism plus catabolism. Anabolism is to build something, catabolism is to degrade something. So you're half right in that catabolic is a part of metabolic reaction, but not only. And the question is about the specific subset of these reactions that break down nutrients. Then you need to specify catabolic. I do. 3.3, I like this one. The second law of thermodynamics states that the disorder in any system is always increasing. In simple terms, you can think about dropping NaCl crystals into a glass of water. The solvation and diffusion of ions is favored because there is an increase in pH, entropy, ions, or stored energy. Based on the first part of the sentence. Entropy, yes, nice, nicely done. There is an increase in entropy. 3.5. 3.5. A, B, C, or D? They also contribute to disorder in an environment by releasing what? That contributes to the disorder of the system in which they are in. A, B, C, or D? Who said A? Who said B? Who said C? Yes, it is C. It is heat. 3.6. That's nice. This is the kind of question I like. So if you weigh yourself on a scale one morning, then eat 4 pounds of food during the day, then the next day will you be 4 pounds heavier? If this was happening, it's really bad. Because every single day you will be 4 pounds, 4 pounds, 4 pounds, and then by the end of the week you will be like that. You know. So will it happen? No, of course not. All of you know it will not happen, okay? So, why does it not happen? Let's say you eat, let's focus on one type of food, ramen, for example. Let's say you eat ramen. You have, if you are able to, you have 10 kilograms of ramen in front of you, and you eat 10 kilograms of ramen. The next day, of course, you will be dead, but let's say you are still alive, you will not be 10 kilograms heavier. Why not? What happened to the ramen you ate? Especially ramen.
Throw up.
Yes, you will throw up. You will throw up. But at the molecular level, and then you will also on the other end as well, but you know, there will all this garbage that we will not digest, but there will be very little of that in ramen. Ramen is just almost starch, right? So most of it will be transformed in the body. What will happen to the ramen we ate? Or bread, for that matter, or rice, is the same thing. What will happen to the food we eat? Why are we not at the molecular level? What is it transformed to?
Starch will be broken down into CO2 and H2O and released.
Yeah, exactly. So this is mostly what happened. Now most of the food we eat will be transformed into carbon dioxide and water, and then it will be evacuated. Plus, you know, fibers and everything, eventual throw up, all these things will contribute to us not getting fatter and fatter every time we eat. Okay, so 3.11. In the first stage of photosynthesis, light energy is converted into what other form of energy? This question is particularly a little bit annoying because actually two answers are correct. So you have 50% chance. A, B, C, or D. Who said A? It is A. Who said B? Ah, this is not chemical. C, no. It's A or D, kinetic.
Why kinetic energy?
Kinetic energy because it is like the electrons will be in an excited state, okay, and then them moving. Electrons will move and they will have a certain kinetic energy because of their movement that will provide energy for photosynthesis of sugar from carbon dioxide. But there was no mention of that in the chapter 3, so that was a little bit a tricky question. Okay, so this is an electrical. So this is the most correct answer. I think is electrical. 314. Your body extracts energy from the food you ingest by catalyzing reactions that essentially burn the food molecules in a stepwise fashion. What is another way to describe this process? Reduction, oxidation, dehydration, or sublimation? Who said A? No. Who said B? Yes, everybody's correct. 320. Arrange the following molecules in order with respect to their relative levels of oxidation. So, the order is 3, 1, 4, 2, 5. 3, 1, 4, 2, 5. From top to bottom, 3, 1, 4, 2, 5. If you have a question, don't hesitate. Okay? I'm going a little bit fast because I want to release you as quick as possible. But if you have a question, don't hesitate. Okay? This is why we are here on Saturday. Delta G is nice. And the question after. And after. 325 is delta G0 indicates the change in the standard free energy as a reactant is converted to product. Given what you know about these values, which reaction below is the most favorable? Is it A, is it B, is it C, or is it D? That should be very easy. Who said D? Yes, it is D. 329 is nice because we need to think about stuff. I will not read the question because it's long, but if you look at the graph figure Q3329, and then you can see that there is X, this is the energy before the reaction, Y, this is the energy of the products of the reaction, and then you have B, D, and C points. And A. And you need to know, well, you need to sort of say what is really the effect of the enzyme catalyzing this reaction. Is it A, B, C, or D? Slightly more difficult question. Who said A? D minus C versus B minus C. Nice, it's not A. Who said B? Yes, everybody is all right. Number 331. That's very, this is a point question. Favorable biological reactions require what to speed up reactions and meet the demand of the cell? Do they require heat, ATP, ions, or enzymes to speed up the reaction? Who said A? No. Who said B? No. Who said C? No. Who said D? Yes. Which of the following statements are true or false? If a statement is false, explain why it is false. Enzymes lower the free energy released by the reaction that they facilitate. Who said it was false? It is false. Yes. The enzymes will not change the free energy release. It will be the same. B, who said it was true? It is true. C, who said it was true? It is true. Enzyme increase the probability that any given reactant molecule will be converted to product. Is true. This is related to 329, isn't it? D, enzymes increase the average energy of reactant molecules. Who said it was true? Great, it's not true. $346. 346 is, I will draw 3 from here. So we are having 5 graphs. Here there is a little bit of a mistake. This is wrong, right? This bar is wrong. Only this one is correct. But we have a square here, we have a square here, 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 and here. And the correct answer is 2, 3, 4, 1. 2, 3, 4, 1. This one is not part of the answer. Did you get this one? Yeah?
No.
No? What did you get?
4, 2, 3, no, 1.
4, 2,
2,
3,
3, nine.
no, 1. So, this you said it was 4.
Because what you say it is for is the A and B have same potential, and first graph, if our first graph is reversed, left and right, it's
No. So you can see that the arrow here means that this is very favorable reaction. A lot of A will be transformed into B. Okay. So this is number 4, right? And which means that what this arrow means that the energy of B will be lower than the energy of A. Certainly not higher. This means that the energy of B is higher. So then it's not spontaneous.
So why fourth graph, A and B has same energy?
Yeah, that's a good point. That's a good point. So what you say is interesting, because then what you say maybe should be, to be perfectly correct, it should be something like this.
Yeah.
Right? Very nice. You are right. So this one here, it's a little bit strange that the energy of B is not so different from the energy of A. So what is happening?
I guess it is English problem.
It's an English problem.
Equivalence.
So the thing is that because B to C is extremely favorable, the whole thing is drawn towards that direction. This is what's happening. Even though the energy of B is not so different than A, you write like this because the whole thing will dramatically go towards C, because the difference in energy between B and C is very big. So if you want, it would have been here, a bigger arrow would have maybe B also. That the whole reaction is drawn to, the whole reaction from A is drawn towards the right because of this reaction here. So even though B doesn't have so much different energy from A, this reaction is still very favorable because B is removed constantly. Okay, but you are right. This is very nice discussion. So it's nice. So I think if you write like this and justify your answer like this, it's also, I consider it also correct. 352. What is Km? A, B, C, or D? Who said A? Who said B? Who said C? Who said D? No, this is C. The enzyme substrate binding affinity. Km is the enzyme substrate binding affinity. 357. I've seen good answers among you in this one, which is the graph that explained the best the effect of the temperature. 1, 2, 3, or 4. Who said it was 1? Yes, very nice. It's 1. 368 is, I find, a nice, interesting question. So you are studying a biochemical pathway that requires ATP as an energy source. But to your dismay, the reactions soon stop, partly because the ATP is rapidly used up, and partly because an excess of ATP builds up and inhibits the enzymes involved. You're about to give up when the following table from a biochemistry textbook catches your eye. And then you have reaction mediated or catalyzed by enzyme A, B, C, or D. Which of the following reagents are most likely to revitalize your reaction? A, B, C, or D? Who wrote A? Nobody. That's not. You wrote A. You are partly true. You are 50% true. If you add ATP, the reaction will go again. But you must add a lot of ATP. And ADP will still be there to inhibit your reaction. So not only you must add ATP, but you must remove ADP as well, because ADP inhibits your reaction. But adding ATP for some time will work. But your reaction still will stop quickly. So this is not good enough to add ATP. Who said B? Why? Why adding glucose 6-phosphate? Why would it help? Because if you add glucose 6-phosphate and enzyme D, in your reaction you will make glucose and phosphate. How will that help your reaction?
Phosphate will attach to ADP and make ATP.
Not by itself. Not by itself. You need something to attach it to it. So then, according to that, remember what you just told me. Remember, okay? Who said C? Yes. 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 C. Creatine phosphate and enzyme A. But it's very funny because if you look at enzyme A, you see that the delta G0 is positive, which is why maybe many people did not choose this reaction. It's not a reaction that is spontaneous. However, if you add creatine phosphate and ADP, what will happen? Explain to us.
The reverse reaction is spontaneous.
Yes.
So it goes all over.
Very nice. So if you add creatine phosphate plus ADP, and you add enzyme A, it will go towards creatine plus ATP. And it's spontaneous reaction. So it means that in your reaction, not only you will remove the ADP, and you will transform it into ATP. So this is the best way to do. Because you will make a lot of ADP because of your first reaction, and as the ADP is generated, you will transform it into ATP using creatine phosphate and enzyme A. And it's spontaneous, it's minus 3 kilocalories per mole of delta G0, so it will help your reaction a lot. So your idea was good, but you need an enzyme to do it. So the enzyme A, of course. So the right answer is C.
Sorry, previous questions B. Explain why temperature has this effect.
Yes, okay. Explain why. Who tells me why?
As the temperature rises, the reaction occurs faster because of the probability of binding increases. However, when the temperature is much higher, the hydrogen bonds will break, enzyme will lose its original shape, and they cannot interact with substrate.
Yeah, exactly. Absolutely. That's exactly what happens. So at the end, when you reach 100 or when you reach 60 degrees or something, you will denature the protein. It will be broken. 413. A newly synthesized protein generally folds up into a stable conformation. All the information required to determine a protein's conformation is contained in its amino acid sequence. On being heated, a protein molecule will become denatured, and here you had a hint for the previous question, as a result of breakage of non-covalent bonds. On removal of urea, an unfolded protein can become renatured. The final folded conformation adopted by a protein is that of lowest energy. 415. It's about neurodegenerative disorders like Huntington and Alzheimer's and Creutzfeldt-Jakob and mad cow disease and crazy sheep and whatever else. Which is the correct one? A, B, C, or D? What is the ultimate fate of these disease-causing unfolded proteins? They are degraded, they bind a different target protein, they form structured filaments, or they form protein aggregates. Who said D? Yes, this is D. What did you say?
C.
C. Yeah, it's true that there is a picture showing of this big filament, okay? But the correct name of these very filament is more something like a filament of actin or something like this. These very strange disease-causing things, they are called protein aggregates. It's not physiological, it's pathological. So they are called protein aggregates. Okay. 417. Molecular chaperones can work by creating an isolation chamber. What is the purpose of this chamber? Who said D? Yes, it is D. Who said A? Who said B? Okay, B. This chamber is used to increase the local protein concentration, which will help speed up the folding process. So, this is not correct, because if you increase the protein concentration, what you will have is that two proteins will start interacting with each other. But what you want is one protein to correctly fold with itself. But if you increase the protein concentration, then you will have many interactions with many proteins, and this is not so good. Okay? So you really want to isolate one protein, wait until it folds, without interaction with anything, and then you get the protein. 421, A, B, C, or D. Specific amino acid sequences, side chain interactions, the hydrophobic core interactions, or hydrogen bonds along the protein backbone. This is about alpha helices and beta sheets. And they are very common, and many proteins have them. Why many proteins have them? Is it because of specific amino acid sequence? Is it because of side chain interactions? Is it because of hydrophobic core interactions? Or because of hydrogen bonds along the protein backbone? Who said A? This is not A. Who said B? Who said C? Who said D? Nice. Everybody's correct. 426. Beta sheets can participate in the formation of amyloid fibers, which are insoluble protein aggregates. What drives the formation of amyloid fibers? Is it the denaturation of protein containing beta sheets, extension of beta sheets into much longer beta strands, formation of biofilms by infectious bacteria, beta sheet stabilization of abnormally folded protein? Who said D? This is D. D was correct. B was correct. What did you say?
Say D.
You said B.
Did you say D?
D. D is correct. Number 432. The alpha helices and beta sheets are examples of protein secondary structure. A protein such as hemoglobin, which is composed of more than one protein subunit, has quaternary structure. A protein's amino acid sequence is known as its primary structure. A protein domain is the modular unit from which many larger single-chain proteins are constructed. The three-dimensional conformation of a protein is its tertiary structure. Which of the following statement is true? There was only one true. Who said it was A? Oh, A is not correct. Who said it was B? Who said it was C? C was correct. Disulfide bonds stabilize but do not change a protein's final conformation. Who said it was D? It's not B. So somebody said it was B, right? B. A was nobody.
A.
A, you said A, okay. Why is it not correct? Why is it not correct?
I think it's correct.
It's not correct. Which of the following statement is true? It is not correct. Why? Why didn't you choose A? Why B is not correct?
What am I asked about?
I'm asking somebody else. You said it was A, right? A was correct, right? Okay.
I choose A, and
it's not correct.
yes.
A is not correct. Why it is not correct? Why disulfide bonds are formed by the cross-linking of methionine residue? What is not correct in this sentence? Why other people did not choose A? Why didn't you choose A? What is the reason of not selecting A as the correct answer? It is? Exactly, yes. Disulfide bonds, they are between cysteine residues. But I can understand why you put a wrong answer, because methionine also contains sulfur. But it's cysteine. 450. The process of generating monoclonal antibodies is labor-intensive and expensive. An alternative is to use polyclonal antibodies. A subpopulation of purified polyclonal antibodies that recognize a particular antigen can be isolated by chromatography. Which type of chromatography is used for this purpose? I explained this one just last week's lecture we had. A, B, C, or D. Who said it was A? Yes, it was A. Affinity chromatography is used.
Sorry, 444. And what is final conformation? Disulfide bonds will affect tertiary conformation, I guess.
Which A, B, C, or D are you at?
This.
The C. No, so the C is like the protein will just, the tertiary structure will not change.
By disulfide
By
bond.
disulfide bond. Disulfide bond will stabilize the tertiary structure, but will not change it. It will stabilize how the amino acids are organized together, but it will not change the structure. Number 453. Which of the following mechanisms best describes the manner in which lysozyme lowers the energy required for its substrate to reach its transition state conformation? A, B, C, or D? Who said it was A? Oh, it's not A. Who said it was B? It was B. B was the correct answer. So if you remember, the polysaccharide chain was a little bit tilted, okay, so that it mimics the conformation of the transition state. A is not true because this lysozyme, if you remember, only bind one polymer of polysaccharide. So there are no two molecules, one single chain of polysaccharide. 455, I will tell you the correct answer from top to bottom. It's I, A, F, D, G, H, C, B, E. That is the correct answer. Now 460 is interesting. It's also something we did very recently. So enzyme T is most likely to be subject to feedback inhibition by what? So T is the enzyme at the very beginning of this pathway that transforms A to B. So by what is a feedback inhibition likely to be? A, B, C, or D? C. Yes, C is the correct answer. C is the correct answer. Everybody was okay? C? Everybody chose C?
I think B.
B. Oh. No, C is more likely because it's the final reaction in the chain before the you can actually see as 3 units. There is this one, there is one branch, and there is a second branch here. So the final product here, that is the final product of this chain before the 2 forks, is most likely to be inhibiting the enzyme there, the final product of the reaction. Of course, it is possible that some reactions may be like this one or this one, but this final one before the fork is the most likely to be used as a feedback inhibitor. Okay, 463 was particularly difficult, I find. The question is long. The RAS protein is a GTPase that functions in many growth factor signaling pathways. In its active form, with GTP bound, it transmits a downstream signal that leads to cell proliferation. So GTP RAS promotes cell proliferation. In its inactive form, with GDP bound, the signal is not transmitted. Mutations in the gene for RAS, this is true, this is very true, this is what happens in cancer. Mutations in the gene for RAS are found in many cancers. Of the choices below, which alteration of RAS activity is most likely to contribute to the uncontrolled growth of cancer cells? Is it A, a change that prevents RAS from being made? For example, a mutation in the gene that codes for RAS. B, a change that increased the affinity of RAS for GDP. C, a change that decreases the affinity of RAS for GTP. D, a change that decreases the rate of hydrolysis of GTP by RAS. Who said A was correct? Nice. Who said B was correct? That's not correct. Why it's not correct? Because GDP bound to RAS is the inactive form. So if you promote binding of GDP to RAS, you actually promote the inactive form. So there will be no cell division. And the same reason for C. C is also not correct because GTP bound to RAS will promote the activity of RAS, but if you decrease its affinity of RAS for GTP, then you will decrease the activity of RAS. However, D, a change that decreases the rate of hydrolysis of GTP by RAS, it's very good because, well, it's very bad, but if you GTP will still be bound to RAS, but because of quick hydrolysis, RAS will be rapidly inactivated. Okay? Sorry, I said it the wrong way. So if you decrease the rate of hydrolysis, it means that GTP will bind to RAS, but if you decrease hydrolysis, RAS will stay active for longer. And it means the cell division will not be controlled. Okay? Who said D? Yes, you are correct. So you understand why D is correct. So you have RAS and you have GTP here. And then this one is, according to the text, they say it stimulates cell proliferation. But then RAS, as a GTP binding protein, can hydrolyze GTP and become RAS with bound GDP. And in this case, it doesn't do anything, okay? In this case, it just doesn't do anything. Okay? So, the question D, a change that decreases the rate of hydrolysis of GTP, so you essentially, you decrease this reaction. If you can even stop it, this is also possible. So you can stop this reaction. RAS will not hydrolyze GTP, and it will always be active. So always cell proliferation will be promoted, and you get cancer. This is what happens. Okay? Is everybody okay with it? What was your answer? What was your answer?
I can't understand this sentence.
Ah, okay. Which sentence?
I'm okay.
Ah, you're okay, okay. All right. Now, ask a question if there is something you don't understand, okay? 464, what is correct? A, B, C, or D? A conformational change is coupled to the release of a phosphate. The substrate on which the motor moves has a conformational polarity. A conformational change is coupled to the binding of ATP. a conformational change is linked to ATP hydrolysis. Who said it was D? Yes, it is D. It is D. (466). The phosphorylation of a protein is typically associated with a change in activity, the assembly of a protein complex, or the triggering of a downstream signaling cascade. The addition of ubiquitin, a small polypeptide, is another type of covalent modification that can affect the protein function. Ubiquitination often results in what? B is the correct answer. Protein degradation. Ubiquitin is a marker for protein degradation. And finally, the last question is 467. And the best way to describe the mechanism whereby the pool of ADP increase and can bind to glycolytic enzyme, which leads to the production of more ATP, this is called A, B, C, or D. C is the correct answer. This is allosteric activation. So ADP binds to a different site of the enzyme, on the glycolytic enzymes. So they activate the enzyme. So this is allosteric activation. Allosteric because ADP will bind to a different site that actually act on glucose. Okay, so how did you do? What's the, who failed? Who failed the test based on your answer?
Like completely?
Like, who gets 100%? Who gets 90%? 80%? 80%, wow, nice. 70%? 60%? So, yeah, I think maybe, I think most between, but it's a lot of questions, okay? So you were pressed by time, so remember the final test will be much lower question. I think we said 40 or something, okay? So if you have a question, please ask. Some of these questions will be back in the exam. Not all of them, but some of them, okay? I will put them back in the exam. So they are good things for you to get points, okay? All right, and I think that's the end for today. I think it's enough. For Saturday, yeah, that's okay. So if you have a question, don't hesitate. And we see each other on Thursday. And then again on Saturday.